we present to you the chemistry of vision. This will be divided into three parts: wavelengths, the eye, and the brain, for a better understanding of how these individual parts work together. This was done by Anand Vera, Ian Prigmore, and myself, Pascal Cannon.
Wavelengths. In vision lies the stimulus input. Light energy goes into the eyes and stimulates photoreceptors. As an energy wave, energy is passed on through light at different wavelengths. Light is electromagnetic radiation traveling in waves. The frequency determines the hue, and the amplitude determines its brightness. The short wavelengths are at high frequency, long wavelengths are at low frequency, and then it's muted at low amplitudes and bright at high amplitudes. Frequency is the number of crest passes per unit of time. Amplitude is the distance from crest to level of Trudeau, and wavelength is the distance from crest to crest. The wavelengths of visible light range from about 400 nanometers to about 700 nanometers. The lens of the eye absorbs ultraviolet light, which we cannot see. There are three types of cones of the human eye. Each type of cone absorbs different light waves of specific frequencies. Long wavelengths are 564 nanometers in yellowish green. Medium wavelengths are 534 nanometers in bluish green, and short wavelengths are 420 nanometers and bluish violet. The more types of photoreceptors an animal has, the more wavelengths it can tell apart. People have four types: rods and three types of cones. The rods are sensitive to low levels of light, but they do not provide information about color. Cones allow us to perceive color. Each type is filled with slightly different opsin proteins, which are sensitive to different wavelengths of light.
The eye. Vision is simply a process in which light is absorbed by proteins in photoreceptor cells, chemically transformed into an electrical signal, and sent to the brain to be translated into an actual image. Photoreceptors are special types of nerves, which are composed of two types, rods and cones. There are many distinct differences between rods and cones. Rods are more numerous, with there being 120 million rods near the front of the retina. Rods function in less intense light than cones, and embedded within its optical discs is rhodopsin, which absorbs a wider range of wavelengths. Rods are more light sensitive than cones, but can't pick up real color and are monochromatic, registering only black and white. They are mostly responsible for peripheral vision and night vision, and they only give basic information: shapes, light or dark, not detailed like cones. They have slower recovery time, which can be seen when you walk into a dark room. It takes a lot longer for your eyes to adjust. And they have less visual accuracy because many of them are joined to one ganglion nerve at a time. Cones, however, are less numerous than rods, and there are about 6 million cones concentrated in the fovea, or center rear of the retina. There are three types of cones: red, blue, and green, which all give us the ability to see in color. They are photoscopic, meaning they need more light to respond than rods. They contain a protein called iodopsin within its optical discs, which fire an action potential towards the brain. They detect fine detail and color, but are not very sensitive and only reach their threshold in bright conditions. And they have a much faster recovery time than rods, as seen when you're outside for long periods of time in the bright sun and you come back inside. There's a big change in illumination, but your eyes adjust a lot quicker than it would walking into a dark room. These are the structures of the human eye. The cornea, transparent, it refracts light, protects the front of the eye. The iris is the colored part of the eye, and it has two different muscles that contract and expand the pupil. The pupil simply regulates light input. The lens is biconvex, refracts light, and is curved on both sides. The ciliary muscles control the shape of the lens and accommodate light. The fovea is the point of focus, most prevalent in birds of prey, and the retina contains most of the photoreceptors. Beta carotene, which is found in plants and fruits, is converted to vitamin A, which is known as retinol, which is why people say that carrots are good for your eyes. Vitamin A is critical for vision because it is used by the retina of the eye. And retinol is converted into retinol, which is a chemical necessary for both iodopsin and rhodopsin, both present in photoreceptors.
The brain is the part of the body where the visual data that comes from the eyes is processed into images. The optic nerve transmits visual data to the brain's visual cortex from the eye, and it does this via electrical impulses. The optic nerve is actually compiled of over 1 million different nerve fibers. The sheer amount of nerve fibers sharpens the images that the brain can produce. The optic chiasm is a section of the optic nerve. It's where the nerve fibers cross. The nerve fibers cross in the way that visual information is processed at the opposite side of the brain. It's similar to the fact that the right side of the brain controls the left side of the body, and vice versa. The optic chiasm enables the crossing of the optic nerves, which gives humans a better perception of depth. The optic chiasm is more pronounced in predators, as the added perception of depth helps them to better perceive a target from a distance. The visual cortex is the part of the brain that is responsible for the processing of visual data into the images that humans see. The primary visual cortex is the very first part of the brain that processes visual information. The primary visual cortex determines the direction and orientation of things in our field of view, helping us interpret an image. The processing of the basic components in the primary visual cortex lays the groundwork for further analyzation of an image in other parts of the visual cortex. Other than the primary visual cortex, there are 4 more parts of the visual cortex, and each part of the visual cortex processes certain aspects of visual information. Some parts even process the same type of characteristic of an image, such as the first part and the 5th part, both determining the direction of objects. And we can't talk about all the parts of the visual cortex, or this video would take way too long. These are the various sources we use in our project. And this is also the end of the project. Thank you guys for watching.